Good afternoon, everyone. My name's Ivan Power. I'm facilitating the seminar today. Welcome to the Westmead Institute for Medical Research seminar on COVID, which we're calling Get the Vax Straight. Thanks so much for joining us this afternoon. I'd like to start by recording that we're on Darug land at Westmead, and we're commemorating and giving our thanks to elders past and present for preserving that wonderful Indigenous land upon which Westmead sits. This time last year, nature threw us quite a curveball. World leaders challenged science and science responded in a really big way. We're really proud that Westmead Institute for Medical Research, or WIMR, as you'll hear it referred to today, has played a really pivotal role in Australia and the world's response to COVID-19. Our researchers have lent on their expertise into diagnosing, testing and tracing the virus, managing patient care, and now working through vaccine development. Before we get underway, some housekeeping. For the next hour, your microphone and camera are going to be switched off. We really want some questions from the floor here today, so hopefully you can see through the Zoom chat function an ability to ask by pressing on the Q&A button. Please ask your questions in there and we'll do our best during the course of today to actually answer those as best as we can. Firstly, just a little bit about Wimmer. Many of you will know this, being really strong supporters of Wimmer, and we thank you for that. But just some background for those who don't. Wimmer is a leading Australian medical research institute. It's the jewel in the crown of the thriving Westmead Health Precinct, the largest precinct of its kind in New South Wales, and one of the three major projects that the New South Wales government is using to bring the state back to economic recovery after the COVID pandemic. We bring together some of the brightest minds and clinical leaders from across the precinct's four major hospitals to deliver real and significant research breakthroughs. It's truly a multidisciplinary research institute which spans 5 research themes and 11 different research centres. They include the Centre for Virus Research, the Centre for Cancer Research, and the Centre for Heart Research, to name just a few. And really importantly, our researchers are practicing clinicians. This direct connection to patients builds an understanding of the most significant health issues affecting the people in our community, and also about how things developed in the lab are then applied in a clinical context with patients. The result is a transformative impact on the lives of people in Western Sydney, across Australia and around the world. We're really fortunate today to have two of Westmead's most experienced and brightest minds being able to talk to us on the topic of vaccines and COVID. We have with us Professor Sarah Palmer, who's a professor in the Faculty of Medicine and Health at the University School of Medicine. Recently, she pivoted her lifelong research in HIV to leading Wimmer's development of a COVID vaccine for the aging, the only vaccine of that kind under development in Australia. Professor Tony Cunningham, who many of you will have seen before around Wimmer, is one of Australia's most experienced medical researchers, especially in vaccine development. He's been working alongside Professor Palmer to develop Wimmer's COVID vaccine. He's also has contributed his expertise to the New South Wales COVID-19 Vaccine Committee, amongst other ways that he's been consulting to governments and authorities across Australia. So Australia has done really, really well to manage COVID-19 relative to many other jurisdictions. But the same can't really be said in many other places. Sarah, what's the latest?
Sure, so thank you, Ivan. We are very fortunate to be living in Australia. Although we have experienced over 28,000 cases of coronavirus infections and sadly over 900 deaths. When we compare the infection frequency of this virus in Australia to the global infection frequencies, we have a very low infection frequency of the virus. And we have several reasons for why the pandemic has been managed so effectively in Australia. The first reason is because we are an island. So we were able to close our borders when we needed to. Furthermore, when people did come to Australia after the pandemic, they were put in quarantine. So people who could be infectious to the community were maintained in 14 days of quarantine. The second reason is that we have been able to learn from other countries that had experienced the pandemic before us, such as Italy and the United States. And we learned what are the measures that really helped us to contain this pandemic, such as social distancing, washing our hands, and wearing masks. And thirdly, there was a consistent message between the politicians and the scientists. So this consistent messaging really helped the population of Australia to understand this pandemic and to practice those measures that are really important to control the pandemic. And so we are very fortunate in Australia compared to the rest of the world that our infection frequencies are so low. If we look globally, there are over 500,000 cases, new cases of infection daily. And there have been over 2.4 million deaths globally. If we look at the United States, they just passed a very serious milestone yesterday. Over 500,000 people have died from COVID-19 in the United States as of yesterday. So we are very fortunate in Australia. But if we look globally, there are some glimmers of hope. And the hope is the vaccine. If we look to the UK, over 15 million people have been vaccinated in the UK. If we look at Israel, over 30% of the population of Israel have been vaccinated, and over 90% of those over 60 have been vaccinated, Ivan. So that's really, really phenomenal. And also in the United States, over 1.7 million people are being vaccinated daily. So with these vaccinations, the infection frequencies are slowly declining, and we see less hospitalization due to this virus.
Sarah, maybe just pausing there for a sec. I'm a father of 4, and vaccination is certainly something that I deal with my kids when they're first born and as they're growing up. We also deal with it from time to time when we're traveling to, generally more exotic places, but it may not be something that we're really conscious of in our day-to-day lives, which is where COVID's actually taking us. Just tell me a little bit about vaccination and vaccines, just so we can unpack that a bit.
Sure. So vaccination is a medical treatment which actually stimulates our immune system to develop antibodies and T-cell response to infectious diseases or other diseases, to actually build our immune protection against these diseases. In the case of COVID-19, the vaccines include genetic material or protein from the virus, which stimulates our immune system to make antibodies against the spike protein. And this spike protein is what's found outside of the virus. And this spike protein allows the virus to actually infect its target cells. So these vaccines help us develop antibodies towards this spike protein. So in other words, we believe that vaccination really helps your body be better equipped to fight and destroy the virus that is causing COVID-19.
And Australians actually, we heard yesterday, Sarah, that and Tony, that Australians actually started receiving the vaccine. I think actually the first one was dealt out on Sunday night, and the Prime Minister was vaccinated over the course of the past 24 or 48 hours as well. The Therapeutic Goods Administration has approved the Pfizer vaccine and also provisionally approved the AstraZeneca vaccine. But some things I hear about, and particularly I was at another family event on the weekend where someone was telling me that they were concerned because of the speed with which these vaccines have actually been approved. And they were questioning whether there were any corners cut in this. You two professionals will certainly understand that process much better than I. How would you answer a question like that?
So these vaccines have been around for longer than you think. The RNA vaccines have been developed over a period of years, and although they've not been licensed in humans before, they've certainly been shown to work in animal models. The AstraZeneca vaccine, which is produced in a chimp cold virus, has been trialled in many human settings before, and a similar virus to it has been licensed for Ebola. So we do have some experience. Now, One of the good things about Australia is because, as Sarah said, disease levels are so low, we've had time for the TGA to do a proper assessment. We've had time to look at the trials go on for longer, so we can look at safety, and also we've had time to see what's happening in the UK and Israel in particular, and it's very encouraging. These vaccines are safe. The RNA vaccine, the Pfizer vaccine, is highly efficacious, 95%, and preliminary data seems to suggest in Israel that the efficacy of 95% is holding up in the field, and so that looks good. The AstraZeneca vaccine is now being used in the UK, and the important thing about the AstraZeneca vaccine is that its efficacy has been shown to be related, and the paper's just been published in the Lancet, to be related to the interval between doses. And if you can stretch that out to 3 months, it's about 82, 83%. So it's not that far behind the Pfizer and Moderna vaccines. So the side effects that people have been concerned about, in Norway, these deaths turned out not to be due to the vaccine. The people who are getting anaphylactic shock is happening very rarely, about 11 in a million, and can be looked after by observing people immediately after the vaccine. So 95% of that occurs in the first half hour after the vaccine, and most of it occurs in people who've got severe previous allergic responses. Ivan.
Thanks, Tony. Maybe just breaking that down a little bit. Australia's got 4 agreements for the supply of vaccines, and thankfully we have coverage for 250% of the Australian population, which is, I think, one of the highest levels of coverage in the world. There's only Canada, I think, that might have higher levels than that. Maybe if you could just step us through, Tony, you gave us a little bit on particularly the Pfizer and the AstraZeneca vaccine. If there's anything more you want to add about those specific ones as we just go through them.
Yeah, well, the Pfizer vaccine, of course, is being bought in from Pfizer in Europe, and the supplies appear to be unimpeded. They're coming. 10 million doses first up, and then another 10 million doses in the second half of the year. And the Pfizer vaccine is being rolled out first to frontline workers, healthcare workers, quarantine workers, and those in aged care, both aged care occupants and the people who look after them. And that makes a lot of sense. We want to, we're not sure whether these vaccines completely prevent against spread. We think they probably will reduce spread, but there's still information to come, and that will probably be obvious from the UK and Israel in the future. Nevertheless, it's really important that we do immunise our healthcare workers to stop them from becoming ill. We had some problems with that in Victoria, and there's certainly been problems with that overseas. And the case for immunising quarantine workers is pretty obvious, and so is that in aged care. So that's the first tranche of vaccines that will roll out. The second one is AstraZeneca, and that's been, we're getting 3 million doses from overseas and producing 50 million onshore, and that's already being produced by CSL in Melbourne, who produce our flu vaccine every year. So they can grow the chimp common cold virus, which in essence is the substance of the vaccine. And we've got even enough doses to spread out into the Pacific and our new neighbours in the future. So I think that'll be good. So the first phase is those frontline workers. The second phase with the AstraZeneca vaccine, I suspect, will be those people over the age of 70, other vulnerable populations, including the immunocompromised, and also our Indigenous populations, and then onto the people between 60 and 70, and then the general population. It won't be rolled out to people under the age of 18 until we see more trials, and we expect those trials to occur in the future. And we may want to talk about pregnant women, because that's still quite a grey area, is pregnant women, Ivan. So whenever you want to chat about that. Now, we talked about storage. I should just say that the Pfizer vaccine, the reason why it's the first phase is because you've got to keep the vaccine very cold, and that means you've got to limit this really to places that have got these special freezers, and that's usually hospitals and laboratories. So the base for the vaccine rollout is hospitals. Westmead is one of them, RPA is another, Liverpool is another, first up, and then there'll be another succession of hospitals. AstraZeneca will be rolled out in a different way. That'll be rolled out through general practice respiratory clinics and probably pharmacies.
And Tony, just quickly, we see on these three that I think the Novavax vaccine, which is the third, and the Pfizer vaccine, the trial size is around 45,000 people. AstraZeneca is around 65,000 people. That seems like a very large trial. So some of the concerns that people were talking about as regards safety and other things, that appears quite a large sample size. Any views on that?
Yeah, I think that what we don't know yet is of course, when you're rolling these trials out, the first thing you want to do is to protect against disease and death. So that means the focus of the trials has been on looking at what happens to severe disease and people being admitted to the ICU, and at the same time also looking at safety. There are groups that have been left out of those trials, and that's the aging. We don't know enough about the aging, but we do know that the immune responses in the aging for AstraZeneca are pretty good. We don't know about safety in pregnant women because they're usually excluded from these trials. So those types of trials are going to be ongoing. That's usual with vaccines. We look at the special groups afterwards. Novavax is another vaccine similar to the others in that it actually has the spike of the protein in the vaccine itself. So it has the protein itself, not the gene that codes for it. And that's actually stuck together in such a way that it simulates the virus particle, but without any of the capacity of the virus to multiply or grow within us. It's just the protein. Also has an immunostimulant in it, and the efficacy has been 90%, and that's also a vaccine which can be stored at fridge temperature, and it's fairly easy to roll out. We won't get that until towards the middle of the year. The phase 3 trials are now being finished off in the USA. We've seen the trials in the UK and South Africa, which have shown this 90% efficacy.
And Tony, just pausing on that efficacy point, as you described, there were some concerns about AstraZeneca efficacy relative to some of the other vaccines. Let's just unpack efficacy for us and how it is we should think about that.
Yeah, well, I think obviously how efficacious a vaccine is depends on how good it is and inducing the immune response. And as Sarah said, there are two arms to the immune response, antibody and T cells. And we don't understand, that's what we're studying, exactly the exact role of each. We do know the antibodies are really important because they interfere with the binding of the virus to the cell. It's likely because of the other studies on other coronaviruses and other similar viruses that the T cells are going to be really important in the aging and also in duration of immunity. So all of these vaccines have been shown to create good immune responses in the recipients. Having worked out those immune responses, then you go on to look to see whether the virus is efficacious in preventing against disease. So one has a placebo group and a vaccine group, And normally we would wait until the end of the trial, 1 year, 4 years in the vaccine trials I've been involved in. Here, it's been done differently because of the urgency. And so there's been a minimum number of cases, usually over 100 cases, and then people stop and look and see if the vaccine is efficacious. In other words, if you get 90 cases in the placebo group and 10 cases in the vaccine group, then that vaccine is 90% efficacious. So that's what it means by efficacy. However, the trial conditions are quite special, so it's important to see that that efficacy is being borne out when the vaccine is rolled out, as in Israel or UK at present. And it is looking the same way. It's looking efficacious, and also we've not seen any unusual safety signals, which I think is really reassuring. Everybody needs to be reassured about that.
Tony, and which brings me to my next point, which would be great getting Sarah to chat about a little bit, the variants, because one of the ways that we've seen efficacy actually come down is it's in the vaccine's response to some of the new variants. We heard initially there was a UK variant, then we've heard that there is a variant, as we call it, the South African variant, and we do know that in the US there's a number of different variants that appear from the mutation of the virus. Tell us maybe, Sarah, a little bit about how viruses actually develop and mutate, and how you think what that means for, particularly for the vaccination process.
So it's really the natural evolution of a virus to change. And these changes usually give an advantage to the virus. It either helps the virus evade our immune system or it helps it become more infectious. And the strains that were identified in the UK, South Africa, and Brazil, all of these strains contain a change in the spike protein, which allows the virus to be more infectious. It actually brings the virus even closer to those cells that it can infect. So if you think of a lock and key, it actually brings the lock and key closer to the cells that it will infect. So that one change has caused all three of these strains that have been identified in the UK, South Africa, and Brazil to be more infectious. However, the South African strain or variant that's been identified in South Africa, it has additional mutations, and this makes this variant to evade our immune system as well. So more infectious, but also to evade our immune system. And all of the vaccines have lost efficacy against the South African variant. And all of the vaccines that we're talking about, all of them have lost some efficacy against this South African variant because it does have these additional mutations that allows it to really evade our immune system and to evade the vaccines to some extent. And there are new variants coming forward in the United States. So what's really, really important is that we see 85% of people, hopefully globally, vaccinated. Because to prevent variants from forming, we need to stop the replication of the virus. The more it replicates, the more it can create variants or new variants. So we have to really ensure that people are vaccinated, not only in developed countries, but in middle and low income countries. And this will be key to getting us back to a more normal life.
So just one thing to add is that all of the vaccine companies are producing new vaccines against these variants. And the RNA vaccines, they say they can do it in 41 days, but all of the vaccine companies have got this online, and we're used to doing this every year for influenza, so it's not new.
Sure, so Tony is absolutely right. All the vaccines are working on a booster so they could really overcome these new variants. But there is a caveat to that. And that is, if we do not stop the replication of the virus, we could have additional variants that may even be more pathogenic. Because one thing we have found with the UK variant is that it is 50 to 70% more pathogenic and causes more disease.
And so, moving on, I think Tony then was telling us a little bit earlier about the side effects, which are an important part of monitoring that you have through trial processes. I know that you have family in the US and in Europe that actually have so far received their vaccination. What side effects might they have experienced? And also talk us through side effects and why side effects might happen.
Sure. So there are minimal side effects typically from a vaccine overall. And I've had 7 family members now vaccinated. And the side effects that they have had were, my one sister had a little bit of fatigue. My other sister said that at the injection site, it felt like a bug bite. She was itching this bug bite. But overall, they've had very minimal effects. And one person within the 7 people that have, from my family, have been vaccinated is my 94-year-old mother. And she's had absolutely no side effects. And she's excited to get her second dose in a week and a half. So overall, for my family, and they've all received the Pfizer vaccine, that they've had very minimal side effects. And that's usually what we expect from vaccines. However, Tony has mentioned that there is also a low risk of allergic reaction. And this happens within the first 30 minutes of vaccination. So that's why you're asked to stay at the site for an hour, just to be sure that you're monitored. And after that hour, you're free to go. So overall, there are very few side effects to vaccines. But these side effects do indicate one thing, and it does indicate that the vaccine is working, that your immune system is responding to that vaccine.
Fantastic. Tony, maybe tell us a little bit, you were referring earlier to the rollout and the different phases. Maybe just step us through that, what we can actually...
Yeah, I've probably gone through this already, but you can see the phase 1A is for quarantine, border workers, frontline healthcare, aged care and disability care staff and residents. And this is Pfizer. Probably over the next couple of months, we're getting obviously weekly shipments from Pfizer. And then we'll go on to, as I said, that's all being done through hubs, which are based around hospitals because they have the freezers needed to store the Pfizer vaccine. And when we go into phase 1B, that almost certainly will be expanded into the AstraZeneca vaccine, which will be easier to spread around the country. And this will be people over the age of 70, because we know that aging people are more vulnerable, reminding you that people over the age of 80 have a 20-fold increased chance of death than those between 70 and 79, 5-fold increased chance of death. Our Aboriginal and Torres Strait Islander peoples, adults with underlying medical conditions and other critical and high-risk workers. And then, as I said before, adults aged 50 to 69 and then our Aboriginal and Torres Strait Islanders, the residual of those. So people who've got nothing wrong with them and are nice and young and fit, going to have to wait till phase 2B, but that'll probably all be done before October. And I didn't go into the situation regarding pregnant women, Ivan, which I probably should mention, and that is that pregnant women are also more susceptible to this disease than others. So we really like to immunise them, and it's difficult to advise because we don't have safety data. We do immunise pregnant women for influenza because we know that there is an increased risk of pregnant women being admitted to ICU, particularly in pandemic years. So the advice is to discuss this with your doctor. We hope that pregnant women are not in the likely exposed group, so it might be better to wait until there's more information. I would not recommend pregnant women in the first trimester being immunised because there is this slight risk of fever, which can be detrimental. But watch this space. There will be more information and advice coming in the future.
That's really helpful.
This rollout means, Ivan, is that hopefully by the holiday season, we can be moving more freely around Australia.
Yeah, and no doubt we'll deal with this, some of the questions that
And one thing I should say, though, we still do not know the extent to which these vaccines will prevent spread. So we're being warned to be cautious. That's gathering together inside and shouting and yelling and drinking and all the rest is not going to be good for spread. To wear masks on public transport and still to maintain our social distancing is important. We're lucky it's summer, and sitting outside at restaurants is fantastic.
Yeah, yeah, totally. Now, you esteemed people are very fortunate to also be actually developing a vaccine out at Westmead there. And it's, Sarah, you'd refer to as a second generation vaccine, which deals largely with the safety of older people. Tell us a little bit about what you're doing, why you're doing it, and how you're going about it.
Yeah. The what I've been interested in for a long time, and teaming up with Sarah on this one, is vaccines for the aging, because we know that vaccine efficacy drops off very much in the aging. Before we had the shingles vaccine, the new one, the maximum vaccine efficacy was only about 55%. Now we hope that the COVID vaccines will be a lot better than that. But it is likely that we will need ways to boost the efficacy of the vaccines in the aging, and particularly to keep that the duration of the vaccine efficacy going for years. And the key to this is something I've been interested in for a long time, and that is stimulating T cells. And so Sarah will go into the ways in which we've dissected bits of the virus to use them to test and stimulate T cells. What I've shown you on the screen here is that we can take these bits of the virus and actually plug them into another vaccine, into the hepatitis B vaccine, and then they'll be processed by the body, and one will get a response not only to the hepatitis B vaccine, but also to these bits, what we call peptides or short proteins, within the hepatitis B particle itself. We call this a virus-like particle vaccine, and it's very similar to Novavax. A little more, a little bit different, but similar to Novavax.
And these bits of proteins and peptides that Tony is talking about come from the coronavirus, so from SARS-CoV-2. And I have a very brilliant postdoctoral fellow in my group who's used a computer program that has allowed us to identify these proteins that are part of COVID-19 that are absolutely important for this virus to replicate, so they won't mutate. So we're really hoping by using these peptides and these proteins that we will develop a vaccine that really the virus will not be able to mutate and change towards.
That's fascinating, Sarah. And I was fortunate to actually meet some of your team the other day when I was out at Wimmer. And the work they're doing is just, it's almost science fiction in terms of what they're up to. It's just amazing. And it's really amazing to think that all that is being done.
It's so true. We're very fortunate to have very, very sharp minds here. And one other thing I do want to say about those proteins is that we have found that when we expose these parts and bits of proteins to cells from patients who have recovered from COVID-19, that their cells do respond to these protein pieces that we have identified. So it's very, very promising as we go forward.
Yeah, we're using them as a diagnostic test for natural infection. Look at the duration of immunity, which is after vaccines and also after natural infection. We're not doing this alone. We have collaborators at the Doherty Institute in Melbourne, at the Queensland Institute of Medical Research in Brisbane, and also at the Translational Research Institute in Brisbane. So we're collecting the Australian minds up and down the East Coast.
Fantastic. Now we've got quite a few questions coming through from the chat from our audience, which I'm going to throw to in just a couple of minutes. I thought I might just before we do that is just to let people know how it is that we can they might be able to support Wimmer in all the wonderful work that it's doing. Importantly, also be able to spread the messages like the one that we're receiving today about the wonderful things that are going on at Westmead and the wonderful capabilities we have in medical research out at the campus there. First thing to do is to actually tell people about what you're actually seeing today, the experience that you've had in listening to Tony and Sarah talk about this, and find ways of actually sharing that as best we can. You know, the objective of us being able to do sessions like Meet the Researcher and also other ways, which we'll tell you about in a second, of disseminating this information is just so that people actually get the facts on an area that sometimes is subject to a little bit of misinformation. So we think it's really important that people talk about what it is that Tony and Sarah have shared with you today. The other bit is to follow us on social media. We're active on all the usual social channels. You'll find us there. You'll find updates on not only the wonderful news that you get today, also news on the vaccine development Tony and Sarah have, but across all of the centres across Wimmer and the wonderful work that's being done there, that has been being done for well over 20 years since Tony actually helped found the Institute many years ago. Another way to do that is to actually come to events like this. Many of them now we have to have virtually, but we're hoping that once this rollout has actually been completed, that we'll be able to go back to hosting you at Westmead, at our wonderful building we have there. And you can see the extraordinary development that's going on at one of Australia's largest health infrastructure precincts and projects. It really is, having been out there the other day, really is quite extraordinary, the amount of work that's going on and the quality of the campus that's being built. And last but not least, there's multiple ways that you may be able to support us financially, even whether it's very small ways through regular giving that you're able to support. Sometimes it may be in things like bequests and other ways, and major gifts that we can find ways to actually find ways to support Wimmer. And last but not least, if you're an employer or a company and want to actually spread messages like this and unpack issues around medical research, we'd love to find ways of working and partnering with you, particularly at this very critical time. We're having experts speak to your staff and to your leadership teams about these matters. We think is really important to getting the message out there for Australia so that we have a very successful vaccination campaign. With that, I'll throw over to questions, if I may. And maybe just one, Tony and Sarah, if I may start with Melissa, that's asked about the virus being less active in hotter climates, whether we actually have any evidence of that.
No. Look at Indonesia.
Yeah, and I would say look in the African continent. We expected this virus would people had expected this virus would decrease during warm months, but that's not what's happening. In fact, some of the worst infection frequencies in the United States happened during the summertime.
The respiratory viruses often seem to be sensitive to humidity, but there's almost an intermediate humidity. And in fact, high humidity and low humidity seem to be okay for some of these viruses. I think the issue, of course, with cold climates is during winter we tend to accumulate indoors, and we now know that aerosol is important indoors for spread as well as droplet spread.
Yeah, when we go inside, we have a 30% greater chance of spreading the virus. So we have to be very careful inside.
And so, Tony and Sarah, it seems like what you're suggesting, therefore, is that the virus, you know, humidity is important, but the proximity of people to each other in closed environments, which of course very often happens more in colder climates when it is cold, particularly in the Northern Hemisphere, that is likely to be a function as regards its spread much more than the temperature that you might actually be.
Yeah, I think that's right, Ivan. I think it's quite clear when you look at the northern suburbs outbreak in the Northern Beaches outbreak, I mean, and also some of the other outbreaks we've had, they've all occurred indoors. Yes. The Crossroads Hotel, the et cetera, et cetera. So this is, and of course, this was originally detected because of the outbreaks in battleships, and that it could be ameliorated by about 2/3 by wearing masks. So aerosols are important. And quite frankly, the thing that scares me most as an aging person is actually going indoors, and particularly I think in the pubs, which where people do tend to lose their inhibitions, scream and shout a bit, and there's droplets circulating.
Another question that's come through from Susanna She's asked, are asthmatics more susceptible to vaccine side effects?
No, there's no evidence that that type of allergy, I know why it's being asked, because asthma has an allergic basis to it. But the severe allergies that people are talking about in terms of predisposition to anaphylaxis are previous things like bee sting, anaphylaxis, etc. So that type of allergic response it's a so-called immediate type of allergy that people are concerned about more than anything else. I'm not an allergist. I know a bit of immunology, but that's actually more of a concern. What is a concern is that people with asthma do have a slightly increased risk of serious disease. Yeah. It's not as marked as the immune compromise, but they certainly do. And the severer the lung disease, the greater the risk.
We've got a couple of questions come through here from people around the different vaccines, and I think it's maybe good to unpack them because it allows a good discussion of that. First one is around, given this is one of the very few viral vaccines, Martha's asked us a question, is it as effective as bacterial-based vaccines?
More so. There are lots of viral vaccines we use in kids, aren't there? Yeah. MMR, measles, yeah. So
hepatitis B.
And you know the
HPV.
Yep. Yeah, absolutely. Hepatitis B is absolutely the sine qua non.
Yeah.
So, and the shingles vaccine I've been involved with adults is 90% effective using a single protein and a good immunostimulant. So I would have said that, you know, some of the bacterial vaccines and malarial vaccines are the ones
not
quite as effective. Okay, diphtheria, pertussis, tetanus, all toxoid vaccines are very good, but when you get down to pneumococcal immunisation, it's not as effective in ageing people as this shingles vaccine, probably because of a whole set of issues I won't go into. One being there are more than 23 subtypes of pneumococcus, which is an issue.
Some more questions here, in particular around the AstraZeneca versus Pfizer. Melissa's asked us why Pfizer's vaccine got approved before the AstraZeneca vaccine. Was there any
quicker?
Yeah, they were approved by other agencies such as the FDA in the United States first.
Yeah.
And AstraZeneca took longer to be approved, and they were quicker out with their product.
Yes,
and
that's
so
right.
Very fast.
Pfizer were first everywhere around the world. Yeah, they even beat Moderna, and that's what happens when you get a couple of great Turkish scientists in Germany linking BioNTech, which we always forget about because they were the guys who did it, and they linked up with an experienced vaccine company like Pfizer. AstraZeneca is not an experienced vaccine company. Interestingly, the other ones that I know so well, GSK and Merck, have not come out with vaccine candidates yet.
And Merck has stopped their vaccine program.
Yeah.
So.
And just in relation, another question that looking here, Robert has asked us, with the AstraZeneca vaccine, how protected will he be after the first shot or the second shot 4 weeks later?
Strangely enough, 70% is the recent article in The Lancet, if you want to look it up, and that seems greater than the first results of 62.5% if you get 2 standard doses. And part of the reason for that is those 2 standard doses were used 6 weeks apart, not 12 weeks apart. Strange. 70%. Within 21 days of the first dose, you start to get an improvement in the efficacy of the vaccine. Within 21 days.
Tony, is there any rhyme or reason as to why that time period?
Well, Andrew Pollard, who's the guy who's pushing the clinic, not pushing, he is the senior investigator, wondered about the certain immune changes. Other people have wondered whether you might get a little bit of immunity to the other part of the vaccine, which is the chimp common cold virus. and that has to decline before you get a better response. We don't know yet. I think that's got to be worked out.
And Tony and Sarah, a question here from Simone, who asks, if you're anaphylactic to bee stings, are you more likely to have an allergic reaction to these vaccines?
Well, that's the concern.
Yeah, it is.
That's the concern.
Yeah.
If you do have had anaphylaxis, then you need to tell the people who are going to give you the vaccine, and you need to be
watched
closely.
Yeah.
But
Nobody has died from these vaccines, and there's been a lot of the vaccines rolled out.
Yeah.
So no one has died from a response to the vaccine, but there have been some serious allergic reactions.
That's why you need the EpiPen
And right
nearby,
away.
you know, right away. Lie you down, and if you start to get faint, boom, in with the EpiPen. It works very well.
Right, and so therefore, people who have some experience of anaphylactic shock, it should not stop them signing up to actually be vaccinated, because
exactly. I think particularly if they're at risk, if they're healthcare workers, you may actually go for one of the other vaccines if you're not in that frontline group. But if you're in that frontline group, then you really need to talk to the people administering the vaccines to you.
And let them know.
Let them know.
Yeah. But that's the reason that they have you stay on site for 1 hour after the vaccination, to ensure that if there is a serious side effect, that will be administered to right away.
Right, and tell us a little bit more about the likely, as I think you were describing, Sarah, about the response to variants that we're getting and mutations in the virus, about the likely use of boosters over time. Do we envisage that you'll be vaccinated, with the 2 doses, and then that's it for the rest of your life? Or do we expect, in terms of the coverage?
Tony and I both believe that there will be boosters for this virus. There may be yearly boosters for all I know. We just have to watch and see how the virus changes over time. And furthermore, what's really key, and I can't stress this enough, low and middle income countries need to have access to these vaccines. If we don't stop the replication there, then these new variants will be formed constantly. And then that will lead to the vaccines being less efficacious, for sure.
You've got to lower the global load of virus, I think.
Absolutely.
But I think this business about will this virus become like there's a lot of debate about whether, among virologists, whether this virus will become benign, or whether we'll get recurrent bouts of quite severe flu-like illness over time. And we don't know the answer to that, but I would be betting that we'll need recurrent boosters just like flu.
Yep, I would agree. For all we know, maybe we'll mix them together and have one shot.
And so just unpacking that point a little bit more, Sarah, coming back to then, as you said, about low and middle income countries, in terms of then international borders being opened, what do you think? It's a question to which there really is no known answer at this point in time. But what do you think are some of the factors that will feed into people making decisions about that over the course of the next year or more, as people are thinking about the risks involved and benefits involved in having borders open, particularly about that point about vaccination levels in low to middle income countries?
For me, I would think that we'd have to have absolute access to low and middle income countries before we can travel. And I think that will limit our travel till 2023 until we're absolutely sure that the majority of people have access to vaccines in these areas. And whether there's been, and then we often have to track how well the vaccines are working in low and middle income countries to ensure that the coverage is just as good as it is in Australia, Europe, and the United States.
I think we'd have to know also what effect the vaccine has on spread.
Yes.
That's going to be the critical issue. I think we're seeing good things in Israel. How much of that's lockdown, how much of it's spread. Okay. And, it's being judged by surrogates in terms of admissions to hospital, which obviously we expect because these vaccines prevent disease. But with the AstraZeneca vaccine, just to remind everybody, that the effect on uptake on the development of infection essentially dropped from 80% down to about 50% in terms of effects on infection. So I do expect there will be some effect on spread. We don't know to what degree, and the studies in the UK and Israel will tell us that.
Yeah, absolutely, Tony. We don't know the magnitude that these vaccines will have on, what magnitude will they stop the spread? We just don't know.
And so absent international borders being open, if we get to your magic number, Sarah, 85% of Australians actually, adult Australians being vaccinated,
right.
Is that a world where we have to have huge precautions around social distancing? Just talk us through what you think that world might look like.
So if 85% of Australians take the vaccine, I think we will be able to open all borders within Australia. We may even be able to open up a travel bubble between Australia, New Zealand, Australian countries that have a high uptake of vaccines.
As long as they don't beat us at cricket.
Oh, I don't know. I'd still like to go. But anyway, so I think it really is key.
Rugby we allow, but cricket not.
It really is key, the amount of vaccination that really takes place globally. So what this really tells me is how dependent we are globally when it comes to a pandemic and actually to containing a pandemic.
Right. And in terms of then the behaviours within Australia, in terms of restrictions on actual and social distancing and the like, you expect with a fully vaccinated Australian public, even absent movement internationally, that we will be able to return much more to a more normal life. And so therefore the importance of getting that 85% level to the economy and to mental health in this country is actually really, really, really critical.
I would totally agree with that, Ivan. I mean, I think if we have 85% of Australians taking this vaccine, we could almost go back to where we were before the pandemic. I would call it the new normal, and we will still have to take some precautions. But overall, because there will be 15% of people who may not take the vaccination, but if we get up to 85%, life will go back to the new normal and close to normal as we had known it prior to the pandemic. Wouldn't you agree, Ken?
I'm usually an optimist, but I'll sound one pessimistic note, and that is that the quarantine issue. And that is that if we get spreadable vaccine evasive strains coming in, and we have already, there've been 11 or 12 of the South African and Brazilian strains in quarantine. We don't know about that. How well have our people done to restrict the over 50 UK strains, these 11 and 12 Brazilian strains from getting out? But they're the risk. And so if we can immunise our quarantine workers, and if that stops them from spreading such strains, then we should be okay. But if they get out, we're going to have to watch very carefully and not be complacent. I think that's, so our public health people will still be treading on eggshells, I think.
I would agree. So we will have to maintain our vigilance and sequencing of the virus as...
Absolutely, yeah. So probably the difficulty also is what happens if people's testing goes right down and people who've got common colds assume they're due to flu, etc. And that makes it much more difficult for our people to trace this. So the testing has been great. Sure, we can still test our sewage and look for fragments of the virus, and that might help us as well.
And just a point there, Sarah, I think is really important. One that I'm fortunate enough, sitting on the board out at Wimmer and being, chairing the foundation to actually see the work that you do on very close quarters. But my observation would be that the new normal that you describe is such a powerful new normal because of the wonderful strides that we've seen in medical science over such a very short period, and an acknowledgement about what are the things that we need to do to be allocating more capital, whether that's money or whether that's actually really smart people, to continue to make that happen. And so arguably we're in a position where we're actually in a bit of, maybe even in a safer world, because we're more aware of some of these risks and we're better prepared to manage them because these risks are always there. And so as some very leading investors in the US have described that the world, the year of 2020 was really the first year of the 21st century, when it is that we're seeing some of the developments that we always dreamed about in terms of, whether it's in terms of technology or particularly in terms of medical research, actually now coming to people. So I think it's, of all the damage that's been done to humans and also to economies and to people, there's some bright lights and also some green shoots that we're seeing that mean that the new normal is potentially.
I would totally agree. It really has revealed the power of our technology and how far we've come, that we can develop a vaccine so quickly and be rolling it out so fast. So it really, and also to the bright minds that we have that we get to work with. That's really, really key. Our students and our postdoctoral fellows who work so hard and put in many, many hours to help us develop these technologies and these tools.
However, we did discuss last night in a national hookup amongst infectious disease researchers that we can do better, that there may be other pandemics coming, and that the public and the politicians forget about pandemics once they've gone. And so we need better national networks. We're not good enough at manufacturing vaccines in this country in terms of sovereign capacity. We're not good at manufacturing, and we're not sufficiently networked. So I think there's a real impetus coming from the researchers themselves to push government at a time when they can see the need to create these networks in the future. I mean, we're pretty good compared with what happened in the USA, where the administration dropped the pandemic preparedness plans of the Obama administration. That must never happen again.
Tony and Sarah, I'm conscious of our time. There's one more question which I can probably deal with as we close off, which is people are saying, as employers and as people, in different areas, how is it that we can getting these messages out to our people? There's a number of questions that have come through about that. I think, as evidenced by what it is that we're actually doing here, we think that one of the best ways is actually encouraging people to seek out information and to seek out information from the experts, whether that is, which will make a recording available of this and of some of the areas through various social pages. We'd love to hear from employers out there if there's seminars that we can be working with them in terms of informing their staff on some of these issues and giving them the opportunity to ask questions, because the only way that we're able to beat this is to actually be beating it with the science and with the truth and with the information that we can get across, and with great stories and people feeling able that that they can ask the questions and get robust and honest answers, which we think is really important.
Let's make one comment, Ivan, that there is a science of the psychology of immunisation, and we have professors of that both in Sydney and Melbourne, and it's very clear what the message is, and the government's taking advice from those people, which it's great to hear. But we're always going to be up against resistance with about 5% of the population to immunisation, another 15% or more who are hesitant, and that's what we have to do, is to ensure that the information about the safety of vaccines and their efficacy, as we have it, as it is rolled out, is adequately conveyed to the public.
And very unfortunately, Tony, on that note, I'll have to both have a wonderful experience. People who'd like to get in contact with Westmead Foundation, with Wimmer's Foundation, please come through our webpage or find us on all the usual social media channels. We'd love to hear from you about how it is that we could engage more deeply with you in whatever aspect that might be, and also how it is that you might be able to spread the good word and support us in some ways. Thanks to all for making the time, and really thank you very much, Tony and Sarah, for your wonderful contribution.
Thank you, Ivan. Thank you, Ivan.
Thank you.